on to the next session. So this is building on yesterday's session with the FDA around standardization, breath analysis, and building a VOC atlas. So first up today, we have Julia from the team here at Owlstone, who will be giving a talk at Volatile Biomarker Discovery and the VOC Atlas. So over to you, Julia. Thanks.
I'm Julia Greenwood. I'm the VP of Research, Products and Services at Owlstone Medical. In this presentation, I'll be introducing the Breath Biopsy VOC Atlas. So what is the VOC Atlas? It's a repository of breath-related VOCs and the context in which they've been found. In the Atlas, you'll find VOCs that Owlstone have shown to be on breath, so that's differentiated from the chemical background, and that have been identified by a comparison against a pure chemical standard. You'll also find quantitative data, so that's the concentration we find on breath. That's growing as we speak and includes data from a wide range of subjects. So why have we built the Atlas? Once you know the identity and the concentration of a VOC, and if your data is accurate enough, you can start comparing data sets and mining that data for more powerful insights. For example, you can perform cross-validation of results by a comparison with similar studies. We also have a wealth of literature references in Atlas. Those can help us understand the context behind a result and give us confidence around the biological validity of a putative biomarker. Atlas can also accelerate the development of breath analysis technologies by providing links between analytical methods, for example, and the VOCs found on breath via those methods. This can help researchers choose appropriate analytical methods to answer particular questions. When building the VOC Atlas, we have three pillars in mind. The first is that the data has to be comparable in order for us to deliver the benefits described in the previous slide. The data also has to be trustworthy, so those accessing Atlas need to have confidence in the data embedded within Atlas to derive value. And finally, the Atlas has to be accessible. We want researchers to be able to use Atlas and to help the field as a whole accelerate the discovery and validation of breath biomarkers. On to the first pillar. So we need our data to be comparable. And we've broken this down by two aspects. First, the identification of breath VOCs needs to be done with high confidence. And second, the concentrations reported on breath needs to be accurate. To ensure high confidence identification of breath VOCs, we follow this identification workflow. First, we generate candidate VOCs via the analysis of breath and blank samples. Importantly, we differentiate breath signal from background contamination. And for those features, we match them against the NIST library. To select candidate compounds, we prioritize those that match against a breath feature with a high NIST match score. We then purchase those high priority standards as single isomers where possible. Once they're in-house, the chemical standards are batched into mixes, and we use the NIST retention indices to make sure our standards don't co-elute. We analyze the chemical standards at multiple concentrations alongside the solvent blank and retention index ladders. Then to create our chemical library, we check the validity of the chemical spectra. So we ensure the signal intensity is above the solvent blank. We check the peak shape and the presence of isomeric peaks. We then select the ion fragments within a spectrum that scale by concentration. This is the method we use to eliminate spurious or contaminating fragments. The retention index is then calculated, and all of this information is added to the chemical library. We then match that library spectra against the breath features of interest. So we match the ions, the ion ratios, and retention time. For each feature of interest, we choose the 5 breath samples that have that feature in highest abundance. An ID is confirmed then when the feature matches against the library spectrum with an SI and RSI score of 800 or more, and with a retention time or retention index match within a predefined limit. So that limit is calculated based on the particular analytical method that's being used. These match criteria must be met in at least 3 of the 5 breath samples tested. So this ID workflow is applied to all of our breath samples at Owlstone, and only those VOCs that pass our match criteria make it into the VOC atlas. So you can see a screenshot of the atlas here with some of the VOCs we've identified on breath. On to ensuring accurate quantitation then. Accurate quantitation is often a term applied quite broadly to different quantitation techniques. At Owlstone, we differentiate between different levels of accuracy. So the top level presented here is validated system level quantitation, meaning that for a given compound, the entire measurement platform from sampling right through to analysis has been validated for its quantitative output. Now, this level is difficult to achieve, especially for a broad spectrum of compounds. The second level down is validated analytical quantitation. This is generally the level we aim for at Owlstone, and it means the quantity reported is an accurate reflection of the levels found in a sample. But in some cases, there'll be caveats associated with the quantities reported. For example, the concentration might fall out of the limits of quantitation. So we currently refer to those instances as unvalidated analytical quantitation. And then the lowest level of confidence is what we call estimated quantitation. So in those cases, we don't expect the concentrations reported to be accurate. One example of this is if your calibration curve has an unacceptable quality. So in Atlas, we aim to report concentrations with validated analytical quantitation. To do this, we have a target panel comprised of Atlas VOCs. This plot shows the expected precision across sequences for that target panel. Now, in this case, we've injected target VOCs onto blank tubes at the low, mid, and high calibrator levels. Obviously, that's in the absence of the breath matrix, and that's one of the big challenges in breath research, is the difficulty in producing technical replicates for the breath matrix. So to deal with that, we've used labeled internal standards for a subset of our Atlas VOCs, and those are injected onto real breath samples. So in this more challenging test, we see that across sequences, we still achieve for most of our targets a precision within 20%. As I said earlier, though, there are often caveats associated with the accurate quantitation. As you see here, the VOC on the right-hand side falls nicely within the calibration curve for all of our breath samples. But on the left, you can see, for this VOC, some of the breath samples have concentrations that fall below the LOQ. So the most important thing here is that those using and interpreting the data are equipped with all of the relevant information so that they can make their own judgment about how the data can be used. So in the latest version of Atlas, we report quantitative data. So that's the median concentration and the upper and lower quartile concentrations that we see on breath. But we've been careful to provide flags that highlight instances in which some of the data falls below that level of validated analytical quantitation. So you can see those examples here. In future versions of Atlas, we'll provide downloadable data. In those data sets, we'll provide information on the accuracy of quantitation sample by sample and VOC by VOC. The intention here is that users can form their own judgment as to how well data can be combined and compared across studies. What I've described here centers around our second pillar, which is that data has to be trustworthy. Another example of how we ensured Atlas is trustworthy is around how we handle third-party literature. In Atlas, we provide literature references. Here, for example, when you search for malaria, we list out VOCs that have been reported in literature as associated with malaria, and we provide links to those literature references. But we know that we can't always be confident in the identity of a VOC reported in literature. So to handle that, we have this ID match column, which tells the user how the chemical reported has been identified. So that will inform the user as to the weight they should put behind the evidence that any particular VOC is in fact related to malaria. In general, we'd assign more confidence to a VOC that's been identified via match against a chemical standard rather than match against third-party library. In the name of producing trustworthy data, we've recently entered into a collaboration with the FDA, a partnership that we're really excited about. They're working on providing regulatory science tools to ensure high-quality breath analysis and VOC identification. So together we can align our processes for producing high-quality data, thus helping ensure Atlas is trustworthy. Our final pillar then is that data has to be accessible. We're in the process of rolling out the initial release of the VOC Atlas. Our intention is to work with partners to integrate their own data sets as well as our own, and to provide downloadable data with all of the important associated metadata, such that external users can analyze and manipulate data for their needs. We intend to provide as much information on the subject sampled and the analytical and sampling methods used to provide as much utility as possible. I'll now hand over to my colleague Nabitha, who will give you a demonstration of the current Atlas release.
I am Nabitha Nagalingam, a principal translational scientist at Owlstone Medical with a research background in microbiology and digestive diseases. Our VOC Atlas aims to assist researchers across a wide range of topics, including microbiome research, by capturing information such as biological levels of microbiome-associated VOCs detected on breath. Inside our body, an unseen community of trillions of bacteria is actively contributing to our health and well-being, our gut microbiome. While these microbes help digest food, their role goes far beyond that. When we eat fiber-rich foods, the bacteria in our gut ferment this fiber, producing short-chain fatty acids like butyric acid and acetic acid. These two acids play a key role in keeping our gut and overall health in balance. Butyric acid, produced mainly by certain beneficial bacteria, is particularly important. It's the main fuel for the cells lining our colon and can also seep into circulation, making its way to the lung to be detected in exhaled breath. Acetic acid is also produced by gut bacteria and is usually more abundant than butyric acid. Therefore, it is unsurprising that it can also be detected at higher levels on human breath. Here, I'll demonstrate how to navigate through our VOC Atlas using these two example VOCs. On the compound overview page, first type in the name of the VOC of interest, in this case, butyric acid. This then returns a page with background information at the top, including certain characteristics and biological roles as documented in literature. This is followed by some biological sources of the compound, and as you may expect, microbiome and diet are listed here. Next, information on chemistry is noted, including the structure and formula of the VOC. In the following literature section, studies that have investigated butyric acid in different disease states are listed and links are provided. This section summarizes information from linked scientific papers, including the disease area, such as colorectal cancer, celiac disease, and IBS. Furthermore, we've also extracted the direction of change reported by the respective authors, the sample source, and the study type. Next, we've provided some graphs that conveniently depict VOC ranges found in populations that our VOC Atlas team has tested, and further info pertaining to these studies. Similarly, for acetic acid, the summary text is displayed at the top, which can include characteristics, sources, related diseases linked to the VOCs. And again, this is followed by a brief description of previously documented biological origins, basic chemistry information, literature resources, and finally, info relating to on-breath levels found during research conducted by our team.
Thank you for listening to our presentation on the Breath Biopsy VOC Atlas. If you're interested in using the Atlas yourself, you can join our waitlist by scanning here or going to the Owlstone website. And of course, thank you to the whole Owlstone team who have created what has been presented here today.
So we've got time for questions, both from the initial talk from Julia and some of the Owlstone team, and Kavita as well. Just a quick reminder to everyone, submit your questions through the Q&A button. Kavita, I might open up with a question that I had for you. So it sounds like, obviously, when we're thinking about specific VOCs that relate to disease, they're dependent on the output of studies, and they may change, as you say. Like the other use cases where a target gas may be known or already identified, but when we want to evaluate or test sensors, what we're trying to do is determine, can you accurately detect and quantitate on that known thing, given the background of all the other VOCs that are present on breath? And that is unlikely to change if we kind of know, what compounds are there, what the reference ranges look like. Is that kind of in your thinking as well for the types of cylinders that NIST would produce?
I think that's absolutely an aspect of our thinking. And it's a really good point that you bring up, that there are a number of compounds that may not be the compounds that are of interest for a particular application, but they're there, they're in our breath, and we can't avoid them, right? And so I think that the goal initially is really to focus on compound classes and understanding which compound classes we can work with effectively. But that's where I say there's information coming from the studies, but there's also the information that we already know that, for example, acetone will be in that breath matrix. And if a detection technology can't contend with that, that's likely to be a problem, right? Because it's probably at a higher concentration than whatever it is that we're trying to detect.
Yeah, exactly. And it's interesting that one of the points you brought up earlier, I think, about the stack gas example. So if not all compounds are present in the background, depending on the sensor or analytical approach, it may or may not be an issue to have certain things in the background. So we know, for instance, when we've done work with metal oxide sensors, carbon dioxide can have quite a big influence. So that minor component that we put into our test rig is very important for those sensors. But actually, maybe for an ion mobility device, it's less important because you're not going to ionize that gas. Anyway, that must make it quite difficult to know what should be in and shouldn't be in, because it does need some knowledge of the likely detection technologies.
And I do think that this is a situation where we will evolve together, right? So we can test sensor technologies with the best information that we have, and then we will learn from those sensor technologies and be able to adjust what we're creating. And right when something is actually at the point of being field deployable, well, then that's locked, right? At that point, whatever those technologies are locked, and whatever's in that mixture needs to be locked too. But I think the benchmarking process, you can draw it as a neat little box, but ultimately I think it's much more iterative than that, because there's a lot of information to be gained from those studies. And I'm actually really glad you brought up the protocol gases, because the point I wanted to make was that we have to identify which aspects must be duplicated, because we will be limited in our ability to add every single possible compound.
Yeah, fantastic. Very last question for me, then we'll open it up to the floor. So where you have the cylinders, so your major minor components, the cylinders provide in a mix, and then when you want to introduce single components, so you talk about the different options there and how the saturator tends to be the preferred approach. I was wondering why permeation tubes were not really in the consideration, because I think one of the advantages that we've seen for them is because they're traceable back to a primary standard through that, measuring the mass loss. we find they're quite helpful to give you nice independent control of a single gas when you want to then change other things in the background as well. So curious as to why the saturator may be the way to go rather than permeation.
So I think permeation tubes and saturators actually offer a lot of the same advantages. I think the key aspect to both of those approaches for adding a single compound is that you have a pure compound. And that's one of the things I really like about both our saturator approach and the permeation tube approach. I will be honest, we have a lot of experience with generating saturated vapors. And we have the vapor pressure data. So partly it is a approach that's based in our 10 plus years of experience, but I don't think that it's out of the question to examine whether permeation tubes would be effective there as well. I think either of those options is a little bit more straightforward than an evaporation chamber that involves a solution in water. Now, the solution in water, right? I mean, it works great for ethanol, and it is a quick approach, but I don't think that it's as easy to deploy in the scenario that we're talking about here.
Brilliant, thank you so much. So next question here, this is probably more for you, Julia. So how have you chosen the list of VOCs to go into the atlas?
Yeah, good question. Thank you for asking that. It's a really important thing to address. So right now in the VOC atlas, the list of VOCs we have are compounds that at Owlstone we have measured on breath. So importantly, that is differentiated from our system background. So we've got different metrics we use to make sure we differentiate breath VOCs from background contaminants. They're fairly stringent metrics, but it's really important because we don't want contaminating compounds to creep their way into the atlas, or at least unknowingly. So they're VOCs that we measure on breath and that we've identified with high confidence. So that means we've verified their identity against a pure chemical standard. But it's fair to say that Atlas it's really in its early days still, it's still growing. So we know there are VOCs out there that are probably on breath that we have not measured to be on breath, or we haven't properly verified yet against a pure chemical standard. So we know there are more VOCs out there that are on breath that are still to come. So in the background, we are developing different analytical methods, looking at different sampling methods, different ways of making sure we cover the broadest possible spectrum of VOCs that do exist on breath, and they will then find their way into Atlas as well. So for now, that is the list as it stands, but it is growing as we speak.
And that kind of last point you brought up there feeds into another question here, which is, does Atlas contain breath from other analytical techniques? So you've talked about, you can have different flavors of GCMS, but again, if you know the identity and you quantitate, in principle, you can have different approaches. So does it include non-GCMS data, or is there any plans to include that type of data?
Yeah, for sure. So right now, it doesn't include non-GCMS data, the version that we have, but there's definitely the intention to include them. If we go back to sort of pillars or the principles that we're using to build Atlas, the key things are that we've got confidence in the data and that it's comparable. So as long as we are confident in the identity that we're reporting, and preferably in the quantity that we're reporting, then in theory, it doesn't matter which analytical method or technique we've actually used to produce that data. So the important thing is that we align on the quality of the data that goes in there, but the process, the technique we use to actually produce it, we are definitely open, and we should be considering all sorts of different techniques that can then go into Atlas. And of course, that potentially then broadens the number of compounds, or the scope of the types of chemicals we can actually then capture in Atlas as well.
Fantastic, thank you. Next question for Kavita. So you touched already a little bit on some of the complexities of having different gases in the single cylinder. There's both stability considerations, And you had the chart earlier that showed, ethanol, hydromethane are quite high abundance, but actually a lot of things are much lower abundance. So I was wondering if you could elaborate a little bit further on how to deal with some of those challenges of those very different concentration ranges and the reactivity of different gases.
So one piece that I didn't maybe talk through in a lot of detail is that it depends on how the gas mixture is going to be used, right? So if ultimately a gas mixture is going to be used straight from the cylinder with no further dilution, then we're in a fairly tricky scenario in terms of having very low ppb concentrations in the cylinder itself. With a delivery system, that actually puts us in a different place where we need a higher concentration in the cylinder. And while higher concentrations are generally more stable, there's a limit to how high the concentration can be in the cylinder before there are other problems that come up associated with the vapor pressure of the compounds. So let's see. In terms of dealing with this, I think ultimately we will need to have the use scenario worked through. So we need to know whether the cylinder will be used directly or with a delivery system. And then there is a way to, it is absolutely possible to put some different concentrations into the cylinders to adjust for the different concentrations that are found in breath. That isn't maybe the first approach that we would take because, well, when you're trying to, fail fast, understand stability, and so on, it's not the moment to make each compound at a different concentration. But ultimately, that could be done to look at the appropriate concentrations for each compound. So I hope that kind of answered the question.
Yeah, that's great. Thank you so much. Yeah. Next question from Julia. How will Atlas account for differences in populations and cohorts?
Yeah, thank you for asking that. So right now in the VOC Atlas, we've got a fairly simple data set and we provide some summary metrics on the expected concentration for our VOCs in breath. But as the Atlas evolves in the next few releases, we'll be introducing downloadable data. Now, when you have downloadable data, that gives you the option to include a whole lot more metadata around the chemicals and the concentrations that we're seeing. So for those data sets that we intend to make accessible to the public, we will include as much metadata as we can about the people or the subjects having provided that data. So that's everything from, for example, BMI or sex to clinical data as well. The idea being that we want to give as much information as we can to the users, and they can then make their own judgment as to how the data can be used. So, for example, if they want to use multiple data sets as a reference data set for a healthy population, they can make their own judgment as to what counts as healthy in that case. And if we are able to include as much metadata about the subjects as possible, they can choose, do they include subjects with IBS in that cohort, yes or no, that type of thing. So our intention is to, collect and provide as much metadata as we can on each of the subjects. And that is our way of basically providing as much population and cohort data as we can. And then the user can do with that what they will.
Excellent, thank you. Next one for you as well, Julia. Will a user be able to upload literature references to Atlas? So in the screenshot there was the literature entries, but will there be a way for users to put their own literature in there in the future?
Yeah, in the future, that's very much our intention. So we've put a lot of effort internally into the literature references. We feel that's something that's going to be really useful for people in the field now. But it's obviously there's a lot of literature out there, and we've probably only scratched the surface, really. So I think this needs to be a collaborative effort. And I think we want to make sure it's as unbiased as possible. So I think it's really important for other people to be able to do their own literature searches and be able to add literature accordingly. Of course, we need to be diligent about the quality of data that's going into Atlas. So we will need to work out some processes around making sure the right type of information is being added in. So that is all part of our current thinking, but certainly we see the value in making sure other people can contribute, and perhaps even when they see a literature reference that's perhaps tagged incorrectly, being able to provide that kind of alert as well. So really making it a collaborative effort to make sure that information in Atlas is usable, is trusted, and useful.
Brilliant, thank you. And there's 2 questions here for you, Kavita. I think the first one you've basically answered. So can headspace be used instead of similar gases? I think that's the saturator approach that you described. And then the next one is, could the volatiles with the same concentrations be absorbed onto a TD tube and then desorbed instead of using a cylinder?
Let's see, that's an interesting idea. Certainly that is possible, right? Volatiles can absolutely be absorbed on TD tubes and desorbed. But in terms of as a method for producing a reference or a breath surrogate mixture, it would depend on looking at, you know, how reproducible that is, what kind of uncertainty there is, and so on. And I know that many people work with TD tubes and have a lot of experience with them. And I think my concern would be that maybe there's a challenge with the reproducibility piece. But that could absolutely be a way to deliver mixtures as well, and is really a way to deliver mixtures when people have loaded samples for their own purposes.
Brilliant, thank you. And then time for one last one for Julia. Is there any preclinical data in Atlas? So I assume this means, you know, headspace, animal model data.
So today we do include preclinical literature references. So when we're looking to find associations between diseases and the VOCs we've seen on breath, we do include all of the sort of preclinical research there. But in terms of the VOC data itself, currently in the current version, we only have breath data. But certainly it's our intention to provide preclinical information as well. We think that's really a key aspect of biomarker discovery. In terms of the sort of vision we have for Atlas, we need to make sure that that data has high quality identification associated with it. But we're conscious that it is difficult to quantify preclinical chemical data in the same way that you can with breath. You can't really provide sort of ppb concentrations. So we know there'll be that caveat there. So we just need to kind of architect Atlas in a way that that's clear and obvious to the user. They don't try and mine the data in a way that you're comparing apples and oranges. But we definitely would like that data in there. It's our intention to include it.
Wonderful, thank you. So we're at time for the session. Massive thanks to Julia and Kavita for the contribution today. Really appreciate it. So thanks again.
Thanks very much.
Thank you.
Great.